Thank you, Margaret. It's a real pleasure to be here today, and I want to thank NCSE for putting on a wonderful conference. I'd also like to thank Dick Feely from NOAA and Scott Doney from Woods Hole for providing several of the slides that I'm going to show you today. Just a quick outline. I'm going to talk about what ocean acidification is, where it's occurring, and what are the potential biological impacts, and I'll be spending most of my time on the biological impacts. So this is the general definition of ocean acidification. It's the change in ocean chemistry driven by the oceanic uptake of chemical inputs to the atmosphere. And it's not only anthropogenic carbon dioxide, it's also nitrogen and sulfur, which can be pretty important contributors in the coastal zone. You may have heard this from denialists, that the oceans will never become acidic, so calling this ocean acidification is alarmist. This is a false statement. Acidification is the process of lowering pH, not the endpoint. Since the Industrial Revolution, we've seen a pH change of 0.1 unit from about 8.2 to 8.1, which doesn't sound like a lot, but it is. This is a logarithmic scale. That's actually a 30% increase in hydrogen ion concentration, a 30% increase in ocean acidification just in the last couple of hundred years. But the rates of increase are important here, and you can see on the blue side of the graph, in the last 420,000 years, there's been very little increase in atmospheric CO2. There's been quite a lot in the last 100 years. In the last 100 years, but we're poised in the next 100 years to see a huge increase in CO2 based on the projections. And what this increase means for ocean pH is roughly in the neighborhood of 0.3 to 0.4 pH unit drop in pH, which is roughly 100 to 150% increase in acidity. This is a huge amount. So where's it going? Kirsten showed one graph of this. This is another way to look at it, but it's essentially going everywhere. And most of the CO2 is limited in the upper 400 to 1,000 meters of water, roughly, where most of our economic activity takes place with commercial fisheries, which is a worry. This slide, the top graph shows, this is the North Atlantic, the Pacific, and the Indian Ocean, and you can see that the penetration of anthropogenic CO2 is almost all the way to the ocean floor in the North Atlantic. So aragonite and calcite saturation depths, the saturation horizon is the point at which calcium carbonate dissolves in the absence of other protective mechanisms. And what you'll notice is this depth varies throughout the world's oceans, but it's quite deep in the North Atlantic, on the order of 2,000 to 3,000 meters, but quite shallow in the North Pacific. And this horizon can be as shallow as the aragonite horizon can be as shallow as 100 meters in the coastal waters of Alaska. The saturation horizons are moving upwards towards the surface at the rate of about 1 to 2 meters per year. And this could have big implications for systems like cold water corals, which is one thing that I study. If you look at the global distribution of cold water corals in pre-industrial times, about 95% of those systems were in saturated water. But if you look at the projections for 2100, 70% of those systems will be in potentially corrosive water conditions. Here's a laundry list of potential impacts. I'll just quickly run through them because I don't have time to talk about all of them. Reduced calcification rate, which I will spend some time on. Shifts in key nutrient and trace element speciation, which Ed will talk about in the next talk. Shift in phytoplankton diversity. Reduced growth, production, and lifespan of adults, juveniles, and larvae. I'll touch on this one. Reduced tolerance to other environmental fluctuations, which could include temperature and induced bleaching, coral bleaching. Changes of fitness, survival, species biogeography, biogeochemical cycles, and the really important one is food webs, which you really don't know much about. There's also reduced sound absorption, homing ability, and recruitment. So the calcification responses vary, and we can see from this slide that the top row are crustaceans, which actually show an increase in calcification under high CO2. The big question is at what cost, and it's an energetics issue. If they're expending more energy to calcify, that may come at a cost to other key processes like reproduction and other things. The urchins, some algae, and some temperate corals show a parabolic response, which means they do well or even increase calcification to a point, but then it drops off rapidly. That's the middle portion of the slide. And then finally, the mollusks actually show really negative calcification response almost across the board. So the big question is, what are the ecological implications of these varying responses from different taxa? And the answer is we don't have a very good idea. A lot of this work's only started in the late 90s and is progressing pretty rapidly now. Tropical corals show a pretty clear trend, a decreasing calcification rate with increasing CO2. And the species studied to date show about a 10 to 50% decrease in calcification. They're obviously threatened by a lot of other things too. The synergistic effects of these things are really not well known. So this is a plot of the pre-industrial aragonite saturation state, and the green dots are locations of shallow water reefs, and Carl Safina talked a little bit about this earlier. But what you can see from this is that when PCO2 was at 280 a couple hundred years ago, almost all of the world's reef systems were in this optimal dark blue category with respect to aragonite. The exception is the area off the Galapagos, which does have coral reef systems, but they're different in some ways. They're more fragile. They're poorly cemented. This is an area of high CO2 upwelling, a natural upwelling. But if you look at the projections for approximately mid-century for aragonite, it looks like most of the world's reef systems will be in similar conditions to what we see in the Galapagos. And other studies have suggested that, in fact, many of these systems could be in a state of net dissolution by mid-century, one of the more pessimistic scenarios. So this is an impact scorecard on the number of species studied. And what I really want to point out is the low number of species studied. And this is where we need to, as scientists, need to do a lot more work, and a lot of this is being supported by NSF and others. But if you look at the calcification response across many of these taxa, it's negative. The exception being coccolithophores, which shows a mixed response. If you look at photosynthesis, there's an increase. Seagrasses tend to do pretty well under high CO2. They're CO2 limited. Could be a potential winner in this pretty negative game of acidification. Nitrogen fixation tends to increase, and Ed will talk more about that in the next talk. And reproductive success tends to decrease across for mollusks and the kind of terms that have been studied. But the main take-home point is that the number of species studied, we don't have a very good idea of how these different taxa are going to respond to increased CO2. Shelled pteropods are another one. These are small marine planktonic snails that are a major food source for a lot of our commercial fisheries. Pacific salmon, pollock, baleen whales eat these things. But some of this work by Jim Orr and Vicky Fabry have showed that these organisms actually start to dissolve while they're alive when saturation state goes below 1. So what are the food web impacts? Almost completely unknown, but this slide shows the dietary requirements for 2 or 3 years in the North Pacific for juvenile pink salmon. And you can see that pteropods make up about 60% of their food supply. So the question is, if pteropods take a big hit and their populations tumble, what are going to be the impacts on a lot of these species that we think are important? So winners and losers in areas that have naturally high CO2. This was a pretty interesting study from an island off of Italy that showed what the normal habitats look like under pH of about 8.2, 8.1. And you can see there's lots of healthy calcifiers. This is the left side of the slide. And seagrasses. But in areas where these volcanic vents were emitting CO2, pH could drop as low as about 6.9 to 7. They saw some very different shifts in the biodiversity and makeup of the marine ecosystem. There were no corals or coralline algae or juvenile mollusks, and the adult mollusks that were there were not doing well, as you can see from the bottom 2 pictures. There was a marked increase in seagrass density. Again, we don't have a very good idea of what these big marine ecosystem shifts are going to look like. But places like this give us an idea of what, of the very few places that we know about, give us an idea of what we may be able to expect. The Pacific Northwest oyster emergency, we have a session this afternoon that's going to focus in on this, the fisheries and aquaculture session. But the hatcheries in the Northwest have been experiencing a failure of recruitment in their larval oysters since about 2005. Larval mortalities have been as high as 80%. So the problem is real. It's here now. I invite you to come to the session to hear from people in the industry that have been dealing with this. We've been on the Hill talking most of the day yesterday to several members of Congress about the problem. But we think it's due to low pH upwelled waters off the coast that are coming into these areas. The global models don't do a good job of picking up the huge changes that are ongoing in the coastal zone. They vastly underpredict for the Pacific Northwest. Biological impacts, I'm just quickly going to run through these. Most of our present knowledge stems from abrupt CO2 and pH experiments, only on single species, short-term incubations, and often extreme pH change. So we don't really know a whole lot about responses of genetically diverse populations, the synergistic effects with other stressors, namely increasing temperature is a big one, physiological and microevolutionary adaptations, species replacements, and particularly food web impacts. These are not easy nuts to crack. So I just have a couple of quick conclusions. And so since the Industrial Revolution, we've seen a 0.1 unit decrease in pH, but we have a lot more in the pipeline based on the emission scenarios that we're seeing. We're probably facing some pretty big changes in marine ecosystems across the globe if we continue down the path that we're on. And the rate of this change is so fast, given the geologic history, that it could be beyond the adaptation potential for many of the organisms that we deem important, particularly the calcifiers. The ecosystem responses are speculative at best. Like I said, the science is very new. We need a lot more work in this area, and I think that is coming. But we definitely need a lot more research on the impacts and vulnerabilities in the species tested. That's probably one of the highest priorities, as is the monitoring and modeling efforts pushed into the coastal zone. Like I said, we've got major underestimations in that area, and those are some of the most productive places for us in terms of fisheries and other activities. So that's all I have. Thank you.
So now we've heard about the physics and we've heard about acidification, and we're going to turn to a part of the ecosystem that is often ignored and not really touched on in these conversations, and that's really the very base, the microbial part of the food chain, a little bit on phytoplankton. And to tell us about that, we're going to turn to Ed DeLong from MIT, from both the Biological Engineering Department and Civil and Environmental Engineering Department. Ed is really the person that we all turn to when we want to know something about the microbial systems in the ocean. He's a member of the National Academy with a distinguished record of really unveiling for us what's going on at the levels much smaller than the pteropods, than the fish, than the oysters, and all of those larger organisms that you'll hear so much more about during the rest of the meeting.
Well, it's great to be here and listen to the panel discussions as well as these presentations to see how our oceans are changing. As Margaret mentioned, the microbial world that we see in the oceans is not so well appreciated because we can't see it, much like we can't see pH and hydrogen ions in the sea. Yet microbes are really important, even though they're not that charismatic. So what I'd like to do is very quickly just describe why we care about microbes in the ocean, what we know about the effects of decreasing pH on their activities, species composition, and so on, and what we don't know. So to take a big step back, but it's important to understand context. So let's think about the context of our planet. We think that the Earth's about 4.5 billion years old, and the best scientific evidence suggests that life is as old as 3.8 billion years. And in fact, that life was probably in the ocean and microbial. So microbes have been around for a long time, shaping our planet. The state of our planet today, the oxygen that we breathe, for instance, is a result of the evolution of some types of bacteria called cyanobacteria, which produce the oxygen that we breathe. About 50% of the oxygen that we breathe comes from the ocean, comes from these microbes. So every other breath comes from these organisms, and that's a really good reason to think about them and what the consequences are if they change. Now, you can think about microbes. They're so abundant. There's more microbes on our planet than there are stars that we can see in the sky. And you can think about them as massively parallel, broadly distributed engines that drive energy and matter cycles in the ocean. From the ocean surface, primary productivity, photosynthesis, and so on, to the conversion of that carbon and the energies that sinks through the ocean into the sediments and so on. So microbes are the engines that drive the turning of these biogeochemical cycles. It's a very fundamental reason to understand what's going on. And you can see that visually, looking at satellites from space and looking at chlorophyll, you can see the changes and the fluxes of how these microbes respond to changes in ocean currents, for instance, and the movement of nutrients, the fertilizers for the surface of the ocean, and so on. So that's a graphic representation of what microbes are doing on the face of our planet that we can see from space. Now, the forests of the sea are microbial, and that's another reason why we want to know what the changes that are happening in the ocean, what the effects might be, and how they affect the prokaryotes, the smaller bacterial-like organisms, or the eukaryotic algae that are responsible for much of the primary productivity in the ocean. Now, Kirsten and John have set the stage well for trying to understand what the consequences are of the rising CO2 in the atmosphere, some of which is absorbed in the ocean. And you can see that at places like Station Aloha, where there's an increase in the PCO2 in the atmosphere that corresponds to a decrease in pH in the same waters. But note one thing, it's not just steady, there's actually variation in pH over the course of the year. In the summer, as organisms primarily produce, they suck down the CO2 basically. In the wintertime, respiration exceeds that photosynthesis, and you get a slight rise in the PCO2. So pH is not constant, and that's an important consideration as we think about acidification in the sea. The other important consideration is that there is natural pH variation in the oceans. So in surface waters, which really is where we're concerned, the variation isn't so great, but if you go just a couple hundred meters down, we get down to pHs of below 7.8, which is the estimated pH that will hit in the 22nd century business as usual. So organisms have been experiencing natural pH variation. What do we know about phytoplankton, these microscopic forests of the sea, in terms of how they respond to increasing CO2, decreasing pH? Well, on the one hand, the elevated partial pressure of CO2 basically is feeding phytoplankton, and in some cases that can enhance primary productivity. And this one graph here is a study. It's actually a culmination of about 18 different cruises that were looked at, and you can see that as there's higher and higher CO2, there's greater primary productivity. That's not true for all taxa and all studies, but it has been observed. The flip side is this calcification or decalcification that John talked about, so that some microorganisms lose their ability to form their calcite shells as the pH drops lower and lower with increasing CO2. And these are organisms like these coccolithophorids, where this has been observed. But it's not that simple, because some species respond in this way in terms of decreased calcification and others don't. And this kind of echoes the point that John made, is that we really need to understand more species and their response to decreasing pH. Now, I just want to show one example of a cycle in the ocean where microbes are really important. That's the nitrogen cycle. Again, microbes are the engines that drive kind of the electron flow, the energy changes that changes elements. For instance, nitrogen. So nitrogen fixation, there's a group of microorganisms that can grab dinitrogen from the atmosphere. It's very inert, but they can grab that and put it into organic matter, and that fuels productivity in the sea. Other groups of microorganisms can take reduced compounds like ammonia and oxidize them to nitrate. Those are the nitrifiers. So let's look at a couple of those processes, starting out with nitrogen fixation. Is there an effect in terms of lowering pH, higher PCO2 on nitrogen fixation? And it turns out there is in some fairly important primary producers in the sea, marine cyanobacteria. So for instance, we see with trichodesmium in a number of experiments that at higher partial pressures of CO2, you get higher amounts of nitrogen fixation. So that's pulling more nitrogen in the ocean, increasing productivity potentially. And that's true for a couple different species. It's also true if there are enough other essential elements around, like iron, for this unicellular cyanobacterium called Crocosphaera, which is another important primary producer in the ocean. Now, how about another process? That was nitrogen fixation. Let's take a look at nitrification, another very important process whereby reduced nitrogen gets converted to nitrate. And that's what algae up in surface waters really need for primary productivity as their nitrogen source. So a recent study has shown in the Sargasso Sea and also in the Pacific that at lower pH levels, you actually get lower levels of nitrification. And this would suggest that as pH gets reduced, even in the next 20, 30 years, that there will potentially be an inhibition of this conversion of ammonia to nitrate, which is a central process in terms of the nitrogen cycle in the ocean. So there are reasons to be concerned and to want to know what these effects might be. This is a similar slide to what John showed, so I won't spend any time on it, but the point is that we really have sampled very few taxa, and so our true knowledge of the responses of individual taxa, not to mention taxa that are present in very complex communities, is really not known at present. Now, I don't expect anybody to read this table, but it's from a recent review, and so it's just a chart looking at all the different kinds of processes that microbes might be involved in and which way they respond, either positively or negatively, to decreasing pH or higher PCO2. This was a meta-analysis of all the data available in the literature so far. And if you go through these data in this paper carefully, what you'll find is that we really don't know a lot yet. There are conflicting studies. It's really hard to be able to pull out the trends right now from the currently available data with respect to the effects of pH on microbial processes and the microbes themselves. So currently, if you look at a couple recent reviews that just came out on this topic, there's really two sides of the coin that you can think about. One is that microbes are incredibly adaptable. They've been around on the planet for a long time. They live in extreme environments. So from a very generalist perspective, we don't think that the fundamental way that biogeochemical cycles are turned by microbes will change. But the finer points may change. There will be winners and there will be losers, and it's not certain we're going to like the outcomes potentially. So the other perspective is that we know that things can change. I've showed you that photosynthetic rates, calcification, two different aspects of the nitrogen cycle are affected by changes in pH. So there's a reason to be concerned and to try and understand what's going on. This just kind of echoes, I think, some of the points that John made, that we really only have a small sampling diverse taxa. That's true of macroorganisms and also true of microorganisms. Lab experiments on cultures are really important to get at the fundamental physiology of how life responds to pH, but we really can't extrapolate that too far to what happens in complex communities. So those sorts of experiments need to be done. We need to know how taxa and communities respond in their natural habitats. We don't have a lot of data on adaptive changes both in individual organisms, in populations or communities, and we need to understand that better. Also, as John pointed out, short timescale experiments don't exactly mimic what's going to happen with pH changes over longer, for instance, decadal time periods. And so we need to get a better handle on that, too, with different experimental approaches. Not only that, and this is really important, we tend to think, I just showed you the nitrogen cycle, and of course, that's just a concept. The nitrogen cycle doesn't exist alone. It's a cycle which turns with the carbon cycle, with the sulfur cycle, and so on. So all the cycles are linked. And in a recent study looking at sensitivities of carbon flux in the ocean, Ulf Ribesal and colleagues looked at responses to other variables, for instance, ocean warming and acidification, and how those two might interact. Now, I just showed you in that red box nitrogen fixation. So acidification increases nitrogen fixation, which means you pull in more nitrogen, and that actually allows more carbon to be fixed. So that actually is kind of a negative feedback in terms of increasing carbon, because it allows more carbon to be pulled down. But we really don't have a full handle on these cycles within cycles, on how the different feedbacks affect one another, which is what we want to get to understand how the Earth system, including its life, responds. So I'm not going to go through the shopping list of things that we need to do. I think John touched on it and I did. But in order to get towards predictability, we need a lot more information, not just about individual organisms or even communities of organisms, but how they function together and all the different cycles which they catalyze. And I will leave it at that. Thank you.